The composition of soil air influences most chemical and biological processes in soils. In particular, the presence of oxygen, carbon dioxide, and metabolic byproducts such as ethylene influence the rates of breakdown of organic residues, the soil color, the availability and uptake of plant nutrients, the growth of organisms as well as terrestrial plants, and ultimately also soil formation processes. While the quantity of soil air is related primarily to the porosity of the soil and to the water content, the composition of the soil air is determined by a balance of consumption and release of gases as well as gas exchange. The atmospheric air is composed of approximately 20% oxygen, 0.04% carbon dioxide, or 414 parts per million as of February 2020. And the atmosphere is also composed of about 78% nitrogen, and it has a relative humidity, which is a measure of how much water vapor is in the air, that ranges from, well, essentially 0 to 100%. It is quite variable. In contrast, the soil air contains much less oxygen and 10 to 100 times the concentration of carbon dioxide, while the concentration of nitrogen is about the same as that in the atmosphere, and that is mostly because elemental nitrogen is not bioavailable. The relative humidity of soil air, though, is way less variable than that in the atmosphere, ranging from 99 to 100%, meaning that soil air is almost always close to water saturation, even in dry soils. All plants need oxygen for respiration. Respiration is necessary for life of microbes and growth of plants. And most of these grow best in oxidized or aerated soil because they need oxygen as the primary acceptor of electrons that are released during the oxidation of the carbon in organic materials such as sugars. If the oxygen concentration is too low, certain elements or ions serve as alternate electron acceptors. However, reactions involving electron acceptors other than oxygen yield a lot less energy than using oxygen. The concentration of oxygen in the air then determines the soil air suitability for plant and animal life. The concentration of oxygen is determined primarily by two processes: the consumption of oxygen and release of CO2 through the respiration of plant roots and microbial metabolisms, and the exchange of these gases between soil air and the atmosphere. The exchange of gases is accomplished primarily through diffusion of gases, but also via convection and as dissolved gases with water flow. Mostly undesirable things happen when the oxygen concentration is too low. We form toxic organic acids, nitrate is denitrified, nitrogen is lost to the atmosphere, available sulfate is reduced to sulfide, and most plant roots cannot respire and grow. The breakdown of organic matter is slowed. Methane may be released under reducing conditions. The low oxygen availability also affects the color of soil, where we have red and brown colored in oxidized soils. Now we have blue and gray colors under reducing conditions. Poor aeration and anaerobic conditions can be induced by various factors, which include waterlogging, compaction, particularly of loam and clay soils, very high contents of clays that swell when wet, which close off large pores which would facilitate exchange of gases, and organic matter decomposition by soil microorganisms that consume oxygen. So we would expect inadequate aeration in poorly drained soils, soils with high clay content shortly after rain or irrigation, deep subsoils in clay soils, in highly compacted soils, especially if they had a relatively fine texture. All living things need carbon, energy, and nutrients. Plants derive their energy from the sun and their carbon from the atmosphere. We call them photoautotrophs. Animals, fungi, bacteria, and humans have to obtain their energy by the oxidation of electron-donating molecules in their environment. We call us and them chemoheterotrophs. That means we must ingest organic building blocks that we're incapable of creating on our own, and we must derive the energy from these organic molecules, just like glucose. In doing so, chemoheterotrophs are also involved in nutrient cycling, where the breakdown of organic materials mineralizes nutrients, which means making them mobile and biologically available. So the mineralization or decomposition rates of organic matter depend on a bunch of things. You need to have water available, you need to have the right kind of temperature, the right kind of pH, and a good quality food. But having oxygen available is essential in what we call aerobic respiration of sugars. Now we've looked at respiration of sugars before. Here I'm going to split this reaction into two half reactions where we have an oxidation reaction and a reduction reaction. And it's the sugar that is being oxidized and it is the oxygen that is being reduced. And here the sugar serves as an electron donor and the oxygen serves as an electron acceptor. One easy way maybe to remember this is that the oxidation is a loss of an electron and that the reduction is a gain of an electron. So the sugar donates electrons which are being moved to the oxygen as an electron acceptor, and thereby energy is being moved and freed up for microbial life. So if no oxygen is available, the respiration of sugars may use nitrate as an electron acceptor, thereby starting a process of denitrification. And denitrification is a microbially facilitated process where nitrate is reduced and ultimately produces molecular nitrogen through a series of intermediate gaseous nitrogen oxide products. Then the elemental nitrogen is lost to the atmosphere, and with it, the economic value of applied nitrogen fertilizers. Now, nitrogen is not the only electron acceptor, but there is a preference for electron acceptors, which puts nitrogen right after oxygen, then followed by manganese, iron, sulfate, and eventually leading to a process called methanogenesis. However, going in this order from left to right, less energy is available or recovered from that reaction. So the preference always is for the electron acceptor that gives you the highest gain in energy. Along with the reduction in energy recovered also comes a reduction in what we call the redox potential, which is one way of how we can quantify what the oxidation reduction status of a soil is. So now that we know how important the presence or absence of oxygen is in the soil air for a variety of soil functions, we may approach the exchange rates required to replenish oxygen by first doing a little calculation on the consumption of oxygen. So let's take a root zone 60 centimeters deep. We're going to consider a surface area of 1 meter squared. And in that soil, we have an air-filled porosity of 15%, meaning that we have some water filling some of that pore space, but there's 15% air-filled porosity remaining. And in that soil, we have microbes and we have roots, and together they consume oxygen at a rate of 0.01 kilograms of oxygen per meter squared per day, and we call this a respiration rate. So we can quickly calculate the total soil volume to 0.6 cubic meters, and from that we can calculate the volume of soil air by multiplying that by 15%. That comes out to be 90 liters of soil air. Now the volume of oxygen in that soil air in the beginning of our calculation is going to be equal to the 20% oxygen concentration in the atmosphere, meaning 20% of the 90 liters is how much oxygen is in that soil air, and that is 18 liters. Now we also know that an ideal gas under a standard atmosphere occupies 22.4 moles per liter, and we also know the atomic weight of oxygen being 16, so that 1 mole of oxygen consumes 0.032 kilograms because there's 2 oxygen molecules. Now we have everything together. We can calculate the mass of oxygen in the soil air as 0.026 kilograms of O2, so 26 grams of oxygen in that soil air. And given a respiration rate of 0.01 kilograms of oxygen per meter squared per day, that means that if there is no replenishment of oxygen, we would run out of oxygen after 2.6 days. So obviously an exchange mechanism that replenishes that oxygen and at the same time removes metabolic gases such as CO2 and ethylene is really important. So there are 3 mechanisms for gas exchange, either as dissolved oxygen in water, as a convective flux, or as a diffusive flux. The first one of these, as dissolved oxygen in incoming water, is because although water molecules contain an oxygen atom, this oxygen atom is not what life can use in soils, but a teeny tiny amount of oxygen, and that is up to about 10 molecules of oxygen per million of molecules of water, is dissolved in water and can be used for oxidation reduction processes in soils. Now, this small amount of oxygen that is dissolved in water is further temperature dependent. So the warmer the soil water is, the less oxygen is dissolved in water. And overall, this first mechanism is relatively insignificant in soils. The second mechanism is bioconvection. And by convection, we mean a bulk movement of air in and out of the soil and thereby exchanging gases between atmosphere and soil air. Now that can happen because maybe there's temperature differences, which means that there's also a pressure difference between soil and atmosphere that facilitates exchange. Or because of atmospheric events. Think about a cold front moving through, having a different pressure in the atmosphere than in the soil, which would mean that we would have a gas exchange. The same thing would happen on a much smaller scale in terms of wind or turbulence effects. The other important part that leads to gas exchange is the infiltration of water. Think of a wetting front of water moving through the soil, pushing soil air in front of it, and thereby facilitating gas exchange at deeper layers in the soil. And transpiration also is a leading cause for convection. So think about plants taking up water through their root system in the soil. Well, that water volume that is moved through the plant into the atmosphere has to get replenished somehow. So that we're creating a negative pressure in the soil, which means that soil air would have to infiltrate to compensate for the loss of the transpired water in the soil system. No matter what mechanism, convective mechanism it is, we can summarize this transport in the following transport equation, which says that we have a convective flux, which is a volume of gas going through a cross-sectional area per unit time, is equal to a factor that is quite similar to the hydraulic conductivity, which now includes the viscosity of air and an air permeability term. So this first part of that equation we can look at as an air conductivity term. And it all gets multiplied by a driving gradient, which here is a pressure gradient. And it's not partial pressure, but it is overall pressure, meaning that the convective exchange of gases is entirely a bulk movement of air that is not in response to deficiencies in any given air constituent, but the whole bulk air as a air mass has to be exchanged for this to happen. So the third mechanism for gas exchange in soils is diffusion. And under most circumstances, diffusion is the most important mechanism for gas exchange in soils. The way that we describe diffusion mathematically is very, very similar to the way that we've done this for convection and also the way we've done it for water movement in soils, where we have a flux that is proportional to a driving gradient moderated by some variable that describes the conductivity. Now here the driving gradient is a driving gradient in concentration. So it's no longer a bulk movement of air molecules with bulk air, but here the movement happens because there are different concentrations in a particular constituent. So the movement of oxygen happens completely independent of the movement of carbon dioxide or other gases in the soil. Now this diffusion coefficient, which here is the factor that describes the ease of movement for the exchange of gases, warrants a little bit more discussion. Now we can look at this as a diffusion coefficient in soil that is related to what we call a free air diffusion coefficient, of how that concentration equilibration would happen in the total absence of solid particles or other phases in the system. And this free air diffusion coefficient happens to be such that diffusion in air is 10,000 times faster than the diffusion in water for both oxygen and CO2 about equally. And then the multiplier in front of it just describes that the higher the air-filled porosity is, the greater that diffusion coefficient in soil, and also that the tortuosity of these pathways plays a role quite similar to the way that we've looked at tortuosity for water flow in soils. So the take home is that water really is a barrier to gas diffusion and gas exchange in soils. And when I say a barrier, it really means that if somewhere along this pathway between the concentration gradients, you have a water-filled pore, that water-filled pore will shut down that diffusive gas exchange. So gas diffusion in water-filled pores stops as soon as they hit a water-filled pores, and now we would have waterlogged conditions.